It's very frustrating when patients ask you questions and you haven't got an answer. We see very complicated cases at Great Ormond Street. We see lots of cases with severe birthmarks. And they can be either brown or red or black or white or all different types of colours, or raised or flat. But they're often huge, so we see patients with enormous birthmarks, up to 80% of their body surface area, or lots and lots of multiple small ones, for example. And they often have many different problems in the rest of their body as well, or they can have. We then have to tell them that they may have problems in the brain as well, and they're going to need a brain scan, and that they may have a risk of cancer. So actually these disorders present with skin disease, but are actually more than skin deep. So at the time I started working at Great Ormond Street, which was now 30 years ago, patients would ask things like, well, what caused it? And some people would have a go at answering that. They'd sort of guess. And I used to say, well, we don't know. And then they would say, well, you know, what's going to happen next? Are they going to get cancer? And we would think, well, we don't really know that either. So it was frustrating. There was no good answers. And actually, I ended up with so many questions. Eventually, somebody said to me, sounds like you've got enough questions to do a PhD. And I said, well, you know, what do you mean? And they said, well, you know, that sounds like a research project. And I suddenly thought, great, let's do that. I got into research as a direct result of not being able to answer questions. I was born in Scotland. When I was about 6, my dad got a job in Brussels. And then I grew up in Belgium. I would say both my parents are actually very, very scientific. Their whole way of thinking has always been very questioning, very into what causes things to happen. Lots of scientific type of inquiry about everyday life. But my dad was also actually my physics teacher at school. So actually I had genuine science education from my dad, which was amazing. When I was a child, I was very good at observing small things, I think. I was good at noticing the patterns of how people walk, how people talk. I used to pick up tiny things about, you know, somebody's eyelid or someone else's foot, and the rest of the family wouldn't have noticed. I remember being fascinated by how flies would fly, and different flies would fly in different ways. So, and I used to sort of copy those in my hand. This fly flies like this. I mean, that's sort of boring stuff in a way, but I think I was good at picking up patterns of biology, probably. And I was very, very interested in bodies and how they worked. So from very, very small, I wanted to be a doctor. And once you actually get into the practice of medicine, there was no time to think. And actually what I love is thinking and puzzles and working things out. I then was lucky enough to find pediatric dermatology, actually, and the very rare conditions in pediatric dermatology. And those were fascinating and interesting, and gave me a pull towards staying in medicine. I came to Great Ormond Street and did a 6-month job when I was very junior. And then I was hooked, because it was obvious that none of these diseases were solved. So I would ask lots of questions about them, and nobody could answer any of the questions I had. I think I got embroiled in it like that. I got hooked. I wanted to be able to get treatments for these diseases, and so I wanted to know what the biology was. I needed to work out what was causing the diseases first. And we started with one of the ones that was coming up to the clinic, which was this condition called CMN, which is lots and lots of big moles. Congenital Melanocytic Naevi. So that condition presents out of the blue without any diagnosis in pregnancy usually. So everything's going fine, and then the person will have a baby who has many huge moles, big, brown, thick, hairy moles on the skin. And this condition has been described since ancient Greek times. For these thousands of years, nobody's known what caused it. And so what I was trying to set out to do was find out maybe what had caused that to happen. I don't think I've ever thought I had a career. I think I had my mission to try and solve CMN and these other diseases, and I had a family. but I don't think I really have ever felt I had a career. I think I've continuously tried to do things to move myself up into a better position to be able to get this done. I was very lucky that people helped me to do very, very part-time work. So I actually had a gap of 6 years where I just looked after my children and worked about half a day a week. But in that half a day, it was brilliant. I came to Great Ormond Street, did a clinic and collected samples. So although I was hardly at work, I was doing a lot of reading, mainly on a Saturday morning when, you know, they would go out. But also I was coming in and collecting samples. So I was building up a sort of research base, which turned out to be a great thing to have done because it got the research started. It wasn't wasted time. The problem was that I wanted to do a PhD, but I wanted to do my PhD. And there wasn't anybody doing this. So I approached lots of people to see if I could be supervised in their lab, and I couldn't find anybody. Eventually I found somebody that agreed to see me just once a month and put his name as my supervisor. And then I could apply for money. And so then I got a grant actually from the Wellcome Trust. And at that point it was just amazing because suddenly I could go into the lab. And I knew absolutely nothing. I literally knew, well, I knew a lot of science theoretically. I had done a good science degree, but I didn't know anything about lab work and I hadn't done practicals for years because by this time I was quite a lot older. So I was starting from scratch, but it was so fantastic because it was finally I could get answers to these questions. Suddenly I felt I could do it. There was a big study set up by my predecessor into looking at what might have caused some of these birthmarks. And actually, I then analysed all that data. We didn't find that it was something they'd done or something they'd eaten or something we were left with sort of nothing, in a way. There certainly wasn't anything environmental that seemed to have triggered it. And at that point, I started thinking more about the genetics and whether it could be something that was genetic. You do have these ups and downs in PhDs, and I had a few major things happen. One was that I'd done a huge amount of histopathology as part of the project, and I came in one day and there'd been an enormous clear out in the histopathology department. I lost a huge amount of work in that area, so that was quite sobering. So I was forced to start to think about the problem again. And I actually at that point then stumbled onto a much better approach because of having to start again. I picked up on an idea that I had had a couple of years before and discarded. What I had been collecting in all those years in the clinic before I really got into the lab was samples of skin and blood. And I often had multiple different samples of skin from the same patient. So if, for example, with CMN, you have all these large moles in different places, we frequently had more than one patch and then some blood. And so I got all those samples out again and I retested using a more sensitive technique. I looked for what was common to those patches in the skin but absent from the blood. And I went through the sequence reading, okay, there was 2 mutations in NRAS. It was this mutation or nothing. And I read all the samples and I had all those data. And then I sat down, it was always the evening because it was the children business. I was there with the data, very excited. And I was then matching it to the key, so which sample was which. And I had a big table. And I actually had my 10 patients down the side and I had all the skin, blood, whatever it was along the top, and I went along putting in the results into this table. And it came out, so these 2 mutations, if you had one of them, every single patch of skin that was affected had that same mutation. If you had the other mutation, the patches of skin had that other mutation. And if it was your blood, not a single patient had it in their blood. Or if it was your skin that was a normal piece of skin, it wasn't there either. And also, if it was in your brain, we had brain samples that I'd collected, it was the same mutation would be in the brain. So we knew it actually caused not only the skin phenotype, the skin disease, but also the brain. So that was an amazing evening. And actually, I sat there looking at that table thinking, I'm the only person in the world that knows this at the moment, which was amazing, you know, really exciting. And then, of course, told as many people as I could, but in that minute, you're the only person that has that discovery. That's a very exciting part of science. The mechanism of a mosaic disorder is that you have a mutation in one cell of the developing embryo or fetus. So when the baby is developing and all the cells are dividing, one of them has a mutation at some point, and it can be any point, and it can be any place. But every daughter cell that comes from that cell will keep that mistake, will keep the mutation. So every cell that comes from there will carry that. And what we then see in the clinic is that there are patches of the body behaving perfectly normally and patches that are not. So in mosaic disorders, we often see, for example, asymmetrical growth. One leg might grow more than the other. Half of the face might grow more than the other. Or you may get all sorts of different types of birthmarks, which are essentially cell populations which have grown more than they should. So either pigment cells grown more than they should, or blood vessel cells grown more than they should. Or you can see, for example, in the brain, that the areas of the brain have folded more than they should, but it's asymmetric. So essentially, what we had managed to see laid out in a table was what was happening in the body in 3 dimensions. This distribution of mutation interspersed with normal cells as well. I was very naive at the time. I thought it was going to solve everything, that everybody would think it was as good as I thought it was. And of course, it's just, you know, it was only one step actually. It was definitely a turning point. And it was actually, who it was a turning point for was the patients, because I was then able to answer that question in clinic. So they would say, what caused this? And I would say, it was a mutation during pregnancy. It wasn't anything that you did. It just happened by chance, and it was in one cell, and then it spread around a bit in your body, and that's why it's come out as these birthmarks. And so it was definitely a clinical turning point for us. But actually, of course, when you tell people things like that in clinic, they all would just move on to the next question. They didn't say, wow, that's amazing, how did you find it? So it's always sobering to go back to clinic, because they always want to know, well, what are you going to do about it? Which is good, keeps us on our toes, no resting on the laurels. So we wanted to get straight onto treatments. You can approach science obviously in a very, very neutral, very, people call it agnostic way, where you do large screens and you look, you know, without hypothesis almost, and that's wonderful and I love it. But if you're trying to solve a particular question where the outcome is a disease therapy, you need to be a lot more focused than that. We're in a rush. We don't have an indefinite amount of time to get this done. Essentially, we knew this now was a genetic condition. It is caused by the same gene in 70% of cases and only 2 mutations. What we have with mosaic disorders, which is also seen in cancer, is genes that are working too much. So they have a small change in them that is actually causing a gain of function. They're actually turned on more than they should be. So what we actually want to do is to dial it down. But you can't completely dial it down. You need your normal NRAS levels. So what we need to do is silence or correct somehow the mutated copy of the gene and leave the normal one working properly. I went along to some people that were in gene therapy. Some people thought it was a great idea, why not have a go? And other people said, actually, you can't do it because those 2 copies of the gene are not sufficiently different. They only differ actually by 1 letter, 1 base pair. And they said it's not going to be specific enough. You're not going to be able to do that. So that was a bit dispiriting for a while, but actually I was beginning to learn at this point that to trust myself a little bit more and at least have a go. And actually when we started doing it, we found you can target 1 base pair, and actually people had been doing this before, not in mosaics, but in cancers. There was data. And that's where we've gone with it. We've been pursuing principally silencing RNA therapies. What I thought might happen maybe 5 years ago, I was thinking, it's good, we're managing to target this, maybe we'll be able to dial it down and we'll have a treatment that we can put on once a week, and maybe it will make the moles flatter, maybe less hairy, maybe less brown, maybe it will make things a bit better. But actually, probably about 4 years ago, I began to realize that it had a chance that it may kill the cells. I figured that if we could get a treatment that was targeted enough, we may start to hit other pathways that were keeping that cell alive. And actually, we could also see it on histology. The cells just sit there. They don't actually turn over. They're not dividing much. So we didn't need a treatment that would dial down proliferation. We needed to get at the pathways downstream of NRAS that would stop that cell staying alive indefinitely. And so we predicted, and sure enough, when we started treating our cells in dishes with the siRNA, they die. That was definitely beyond my wildest dreams. I was hoping we could improve this condition. I didn't think we could cure this condition, but I now do. So statistically, I'm told that at this point in a drug development process, you have a 10% chance of getting your drug into the clinic. So that's the statistics. And I'm very happy to accept that. But what I'm not happy to accept is that we might only have 10% chance of getting a treatment, because we've learned so much that if there's something slightly wrong with this one in some way, maybe it's not quite effective enough, or maybe it's not particularly safe in the preclinical testing. We're not expecting that, but you never know. Then we will just be going back to the drawing board and working something up again. So I don't see my overall chance of getting a therapy into clinic as 10%. I see it as better than that. I think we can't be judged on what we've done until we've finished, and I'm fine with that.